Good afternoon, and welcome to another Research in Action brought to you by the Division of Research at Florida Atlantic University. My name is Karen Scapinato. I will be today's moderator. With that, it is my great pleasure to introduce Dr. Joshua Voss, who is an associate research professor at FAU's Harbor Branch Oceanographic Institute. And he's also the executive director of NOAA's Cooperative Institute for Ocean Exploration, Research and Technology. Dr. Voss completed his bachelor's degree at Elon University in North Carolina and then earned his doctorate degree at Florida International University in Miami. His primary research interests include coral reef ecology, coral health and disease, molecular ecology, and marine conservation and management. With that, I'm going to turn it over to him so he can tell us all about coral reefs.
All right, so today I'm going to focus on essentially the work that we've done that's very much trying to take research approaches and link them directly to conservation and restoration of coral reefs. It's a huge effort. It's been a major problem to address throughout my career. I grew up in Florida and have seen changes to our coral reef ecosystems. And so this is an issue that I've seen happen with my own eyes and I'm very connected to as well. So like most of the research faculty at FAU, my team is really the key to our success. I'm fortunate to have a great group to work with, predominantly graduate students in our integrated biology program and our biology and marine science master's programs. I also have several great colleagues and staff here at Harbor Branch that helps us support our field activities and have collaborated on several different projects. And then in order to achieve the conservation goals, we really work very closely with management from the outset in order to define what the objectives are going to be so that we're not only conducting research that's interesting and important, but also going to provide the information or provide the actionable information that will allow managers to make a decision and move forward with that decision with some confidence. Most of the work that I'll be presenting today has been funded either through NOAA, both through the Office of Ocean Exploration and Sea Grant and sanctuaries, as well as additional local funding through DEP and FWC. And we've partnered quite a bit throughout Southeast Florida as well. So I'm hoping that many of you have had a chance to be on a coral reef ecosystem at least once in your lifetime and hopefully multiple times. We're fortunate to be in an area that has some really interesting coral formations, particularly when you start to get down into the Keys. And most of us, when we think about what a coral reef ecosystem looks like, it's this colorful habitat with lots of rugosity and topography that supports really diverse biodiversity and fisheries. And so this is a great example of that from diving last week in Bonaire. Here's another shot as you start to get a bit down deeper in Flower Garden Banks National Marine Sanctuary, areas of really incredibly high coral cover, approaching over 80% in many different locations. And this is what we would want to see. These are the kind of coral reef ecosystems that are productive, thriving, successful. Unfortunately, many of those are starting to look more like this. Signs of disease, signs of bleaching or losing their symbionts and loss, and in some cases transitioning to low coral cover habitat and being replaced by fleshy macroalgae, particularly in areas with high nutrient availability and low amounts of grazers. So the real questions for our research is, what are the major factors that are driving these changes and phase shifts from high coral cover habitats to low coral cover habitats? And what can we do actively to potentially reverse this trend? What are some steps that we may be able to take both at a small scale level and at a broader level to help address these losses and help to protect these marine ecosystems and their ecosystem services? And really the key to the success of coral reefs is their symbiosis. So corals are a cnidarian animal, kind of like a colonial anemone that builds a skeleton. And then inside themselves, they host symbiotic dinoflagellate algae. And it's that symbiosis that really drives the success of corals on a broad scale. So the algae are getting nutrients and protection from the coral, and then the corals are benefiting from the photosynthesis that those algae are doing, getting all kinds of sugars that fuel their growth and drives productivity, or fixing carbon through light energy into the system. And that symbiosis really drives the kind of productivity that we care about from an economic perspective. So many coral reefs are important fish habitats and support robust fisheries throughout the world. They're very high centers of biodiversity. This is also true whenever you have kind of several benthic organisms that are all competing for space. You tend to see relatively higher biodiversity. Here in Florida in particular, where we have both tropical and temperate species, we have areas of reef that may have more than 250 different fish species located on them. Also really important in Florida, coral reefs are key centers of coastal protection. So these reef systems that essentially extend from Martin County all the way around through the tip of South Florida and throughout the Keys provide great ecosystem services in terms of absorbing wave energy during major hurricanes and storms. It's not an exaggeration to say that without some of these reef ecosystems, many, many more homes and businesses would be lost in Florida during major hurricanes. They also support a really robust and thriving tourism industry here in South Florida. We have individual reefs that get more than 1 million visitors a year for fishing and diving. And much of the Florida Keys is around this idea of going to be on or coral reef adjacent in some kind of recreational activity. And then again, when you have lots of organisms all living on the bottom competing with one another, it's common for them to develop secondary metabolites or chemical warfare. And these chemicals that they're producing can also potentially be used for pharmaceutical approaches in humans. I'm not going to talk too much about that today, but Amy Wright, Shirley Pomponi, and Esther Guzman at Harbor Branch all do some really interesting work in this field. So if you look at all of these ecosystem services combined in corals, in terms of their intrinsic value, it's estimated that coral reefs worldwide are somewhere around 13 trillion. There's probably in the neighborhood of 8 to 12 billion in value in Florida, and about 3 to 4 billion each year in revenue, supporting almost 70,000 jobs every year in Florida that are directly related to coral reef recreation and fisheries. So we have all these amazing symbioses supporting these amazing ecosystem services. However, there's several threats to coral reefs that we're facing, and severe concern about the longevity of coral reefs and the longevity of those ecosystem services here and throughout the world. So climate change is obviously a major issue, and one of the biggest factors that it's driving in corals is thermal stress can lead to loss, but thermal stress can also lead to increased storm activity that affects our coral reefs. Throughout South Florida, we do a lot of coastal development. A lot of that coastal development comes along with needing to maintain beaches, and beach renourishment projects, if not done carefully and effectively, can potentially threaten coral reefs through sedimentation as well. Ships running aground, anchors being dropped on reefs have been a previous problem, I would say, and somewhat ongoing in Florida. Thankfully, educational efforts and the rise of GPS on every boat just about has really reduced some of these larger impacts. Excessive fishing can cause all kinds of trophic cascades on reefs, leading to changes in overall reef ecology. So managing fisheries is important. One of the unique things in Southeast Florida is that we have potential threats from estuarine discharge, particularly managed estuarine discharge, since we've replumbed much of our state to make favorable conditions for water management. When fresh water is released out of things like Lake Okeechobee and goes through our estuaries and out onto the reef, these freshwater events can potentially not only expose corals to low salinity, but also bring along with them excess nutrients or pollutants as well. And then lastly, one of the major threats that we've seen expand over the past 40 or 50 years has been a dramatic rise in coral disease. And I'll talk specifically about one of the most recent coral disease outbreaks we've seen here in Florida and some of our efforts to combat that. So overall, our approach to try to address coral reef conservation and research is to combine three things. The first is exploration and monitoring, finding coral reefs, understanding how they function, and looking at how they change over time to try to understand what their trajectories are and what things may be impacting them. But often to understand the functionality or the causality of changes that we may see when monitoring, we need to combine that with controlled experiments where we can look at what are some of the actual stressors that are defined as causing coral loss. So the controlled experiments are really helpful for that. And then lastly, we've been developing a number of advanced molecular tools to try to understand how corals may be related to one another, how they're persisting and spreading at large scales and small scales, and potentially identifying corals that may be more resistant or resilient to certain stressors. And so using this kind of three-part combined approach, there's really kind of three different things that we're hoping to get out of that. The first is innovative research and data products, things that we can share with others to improve our understanding of reefs and come up with ideas that may work to help protect them. Number two is making sure that we directly provide that data and recommendations to the management agencies to make sure that they understand that if they make a decision, they can defend it with some data that is robust. And that it also makes it easier for them to potentially invest in things that may be controversial if they have the data that supports that it's a worthwhile investment. And then being in our role as FAU, I'm really dedicated to and think it's very important that we are training students and doing effective outreach to make sure that the research that we do and the information that we provide, number one, passes on to the next generation of scientists long after we're gone, and number two, helps to inform the public in our region and beyond. So we've done a lot of exploration. We've been very fortunate over the past roughly 12 years to be supported through the NOAA Cooperative Institute. And during that time, we've led 46 different research expeditions, primarily focusing on areas that were either undescribed coral reefs that we suspected might be there, or underdescribed reef habitats, areas that we knew were there but weren't really well characterized. All of these have been collaborative efforts with NOAA sanctuaries or fisheries and several other academic partners. And the outcomes of this have been several things. First, finding those new reef habitats. Essentially everywhere that we've gone looking for new deep reef habitats, we've found them. We've been able to identify several new sponge species and several additional fisheries resources associated with these reef habitats. And then the other major goal is to provide recommendations to management agencies and councils for which areas should be protected and why, trying to promote better coral conservation and management. So in these approaches, we get to use a whole variety of different methods and a lot of fun toys, so to speak. We use multi-beam bathymetry, which is essentially like sonar, but instead of a single ping going down to the bottom and back to tell you how deep it is, it's multiple pings at different frequencies all going at the same time. So you end up with a 3D picture of the bottom rather than just a depth. We use a lot of remotely operated vehicles that are tethered to ships and give us the capability to see and collect in deeper areas. We do quite a bit of technical diving and shallow scuba diving as well. And I'd still argue very, very much that there's nothing that can replace a diver's eye or a diver's hand in the field to observe something or to sample something with dexterity and finesse. We also use things like gliders, submersibles, landers, etc. And first major take-home message I want you to leave you with today is that the coral reef story is not all doom and gloom. As we start to look for more of these deep and mesophotic or middle light reefs, we found that they're broad and extensive throughout the Caribbean and the Gulf of Mexico. Essentially, every place that we've gone looking for mesophotic reefs, we found them, and they seem to be fairly robust and healthy. Rates of disease on these reefs are very low. Coral cover in some places, again, could be over 80%, like in Flower Garden Banks National Marine Sanctuary in parts of Cuba. So it's really more the shallow reefs that are facing a lot of these threats, and at least for the current time being, some of these deeper and mesophotic habitats seem to be demonstrating robustness and resilience in the face of stressors that are affecting shallow reefs. So in flower garden banks, we've done 16 expeditions so far and really focused on doing a lot of benthic characterization. And all of this was with the goal of expanding Flower Garden Bank's National Marine Sanctuary. And that was successfully achieved just this past year, where Flower Gardens expanded from an original 3 banks to now 14 more banks, so 17 in total. And that represented a roughly 300% increase in the total amount of area protected within that marine sanctuary. So it was a huge achievement by the sanctuary program, and not just us, literally dozens of other research teams all trying to identify and characterize these habitats to build the case that they were unique and worthwhile protection from things like oil, gas, fishing, etc. This project also gave us the opportunity to really develop a fairly robust technical dive team here at FAU Harbor Branch. So we've recently advanced into closed circuit rebreathers, and just recently in this past week, added mixed gas capabilities to that team as well. So essentially it allows us to do scuba dives down to 200 feet safely, successfully, and for long periods of time. Also through this project, we were able to test one of the first flyaway telepresence systems. This was developed by our partners at Global Foundation for Ocean Exploration. So they built a system, whoa, sorry, advanced on its own. They built a system that allowed us to stream live images like this in real time off of a ship and to literally thousands of people around the world that were tuning in. And we got excellent feedback and communication from those that were watching. And one of the things that was most impactful to me was that we can generate a whole lot of data about why something is special and should be protected. But giving stakeholders and the interested public the opportunity to, in real time, see these habitats and interact while we're on these habitats can be probably the single biggest factor in driving motivation and effort to conserve a particular place. And I think that this has helped quite a bit in flower gardens. Here, more close to home in the Florida Keys, we've also done a lot of work with both ROV and technical diving to characterize some undescribed mesophotic reef resources, particularly offshore of Key Largo. We identified an area that had recently been multi-beamed by NOAA Fisheries, but had not been confirmed. And through both ROV and technical diving, we were able to identify a very large area, at least 8 kilometers long, of contiguous reef. And that, in an area like the Florida Keys has been heavily impacted by several things over the past 15 or really 30 years. Finding new reef habitat like this was really important and exciting. But there was evidence of impacts even there. There were no impacts from sooty coral tissue loss disease, which I'll chat more about in just a bit. But there were evidence of impacts from fisheries. So we saw bandit reels, we saw abandoned lines, we saw a few fish traps in this area, suggesting that additional fisheries regulation in that area could potentially help to contribute to preserving that fishery and also to preserving the benthic habitat as well. And so this data helped to contribute to the new Florida Keys National Marine Sanctuary Management Plan that has just recently been implemented and is being stood up now. So beyond these kind of site-specific locations, as we've started to do this kind of exploration in a systematic way, one of the major focuses has been to understand how reefs are connected to one another locally and regionally across this great expanse. So I talked about flower gardens, I talked about the Florida Keys, but we've been to several other locations as well, where we've been able to sample corals at multiple different depths to build a comprehensive picture of how are corals connected in a regional space. And what kind of populations do we need to protect in order to help corals to persist in the long term? Are there source populations that are more important than others, or sink populations that may not be that important to protect, or at least might be more vulnerable than others? So this has been an ongoing project with two of my grad students, Alexis Sturm and Ryan Eckert. And as an example of these kind of data, here's one species. This is Montastria cavernosa. It's a star coral that's ubiquitous and common throughout all shallow and mesophotic habitats in the tropical Western Atlantic. And we used very high resolution DNA markers through an approach called 2B-RAD, which essentially identifies differences in the individual base pairs within a molecule of DNA. And what you're seeing here on this map is a series of panels, and in each panel, the left-hand side is a shallow population denoted by an arrow facing up. On the right-hand side is a mesophotic population denoted by an arrow facing down. And then the relative contribution of the colors to each of these individual bars, and each bar represents a coral, tells you which of the coral populations that individual is most related to. And what you can see is that we have some distinct coral populations for this species across the Gulf of Mexico and the northwestern part of the Caribbean. There are some areas like Flower Garden Banks where the shallow and mesophotic populations are completely identical, demonstrating very high connectivity between those two areas. But in other places, we see strong differences between the shallow and mesophotic populations. Places like off the northern coast of the Yucatan, the shallow populations are quite different from those that are further down. And so this suggests that there's variability to the relative amount of connectivity and the relative potential for these healthy mesophotic populations to help reseed shallow areas. In areas like Flower Garden Banks, it should be relatively easy for mesophotic populations to help shallow populations to recover. But in areas like Southeast Florida, that may be more challenging. We may have differences that are fairly stark across sites. It also is quite interesting that we're seeing some connectivity from way down in Mesoamerica near Belize all the way up into Cuba and then the southern portion of the Florida Keys as well. And it suggests that within a generation or two, we may be seeing corals that are providing larvae that get from Mesoamerica to the west coast of Cuba, and then in the next generation from the west coast of Cuba all the way up to Florida. So this suggests that if we want to try to preserve and allow coral populations to recover and persist in Florida, that international management networks may be a really important tool, that upstream areas outside of our national waters could be important sources of larvae that allow corals to thrive here in the long term. So we have this kind of broad picture that mesophotic reefs are amazing. There are variable connectivity across the Gulf, but there seems to be some mechanisms for resilience and recovery. And that's important because the shallow reefs, particularly those here in Southeast Florida and the Florida Keys, are really getting hammered right now. So in 2014, a new coral disease emerged near Miami-Dade and right near Government Cut. It's been termed stony coral tissue loss disease, or SCTLD, or sometimes SCTLD for short. At this point, we know that it's highly transmissible. It's transmissible through the water column. It's transmissible through sediment. It's even transmissible through ballast water, but the pathogen remains unknown. There's been several labs working to try to identify the pathogen, but they've not had success yet. What we do know is that there's been really devastating impacts from this disease throughout Florida and now spreading into the wider Caribbean. What makes this disease so bad is that it has an incredibly broad host range. At least 23 different species of corals are susceptible to this disease. and really fast, strong impacts on corals when it gets to a reef. So we've seen individual reefs with as much as 80% mortality after a stony coral tissue loss disease outbreak. And it's really led to essentially the local extirpation of things like pillar corals in certain parts of the Keys. So it's been a devastating disease, so much so that we started to think about what ways could we try to combat this disease in real time. So one of the things that we initially wanted to look at is why are some corals more susceptible to disease than others? There's variable susceptibility to this disease across species and even within species. On a reef, you can see two conspecifics, same coral species, right next to one another. One may have raging lesions while the other one looks completely healthy and fine. And so we wanted to look at these patterns to try to understand if we could predict or potentially even change something about the susceptible corals to make them more resistant or resilient. And this with the idea of how do we manage these populations and potentially do restoration in the future as well. So working with a large group of collaborators from Nova Southeastern University, University of Miami, George Mason, University of Florida, et cetera, we essentially have what we're calling thrown the kitchen sink at this project, where several different groups are all using their individual expertise to look at what might be different about these more susceptible and more resilient corals. And so our group has been looking specifically at potential differences within the coral genetics for those that are more susceptible and less susceptible. And Ali Klein is a grad student on my team that's been leading this work. And so what we found is that essentially genetic lineage is just not a great predictor of coral disease susceptibility. If you look at this graph here, corals that are more similar to one another are all grouped out together. Those that fall below this red line are corals that are clones of one another based on comparing them to technical replicates. And those that are in red here are the ones that are relatively more susceptible. Yellow are kind of moderately susceptible, and then green are low susceptibility to this disease. And you see that there's not a real strong pattern across this range. There's not a single lineage that's all resilient to SCTLD or a single lineage that's all susceptible to SCTLD. So that means that genotypes is probably not a great predictor of whether or not a coral is going to be resistant, at least within this one species of star coral, Orbicella. So we still hope to identify some things within corals that may make them more resilient. And in the meantime, we've been trying to come up with individual treatments that may be successful in slowing down these diseases, at least to a certain extent. So another grad student of mine, Aaron Schilling, in partnership with Ian Combs, who was a recent master's student of mine, we worked on trying to assess the effectiveness of two of these potential treatments. So a colleague of ours at Nova, Karen Neely, and at Keys Marine Lab, Cindy Lewis, have been trialing a few different things. And they gave us the idea of using amoxicillin with a silicone acetate base called Base 2B, made by a company called Ocean Alchemists. And we tested that against a chlorinated epoxy and went out into the field to treat individual corals and see how they responded. And I was very, very pleasantly surprised and honestly, like truly surprised that when we used that Base 2B plus amoxicillin, that it was more than 95% effective at stopping these stony coral tissue loss disease lesions. So an individual lesion can be stopped with the application of an antibiotic. However, that did not necessarily prevent that coral from getting another lesion in a different location later on. So that means that you would potentially need to do maintenance treatments where you would go back to some of these corals time and time again to do applications of the antibiotic to slow disease. And there's several teams now that are doing that and doing that with success throughout the Florida Keys and in Dry Tortugas National Park. And to help with Dry Tortugas, just a few months ago, at the end of 2021, we partnered with Karen to do an expedition in Dry Tortugas National Park, where we really put a talented team of 8 people. 5 of them were our group, on a boat in the dry Tortugas with nothing to do but treat corals every day, all day long. And with that effort, in 9 days, we were able to successfully treat more than 6,000 coral colonies in the dry Tortugas. Since then, the Tortugas National Park staff have continued to follow up on this, and they've treated an additional 5,000 corals since this initial expedition last year. And so when you take a dedicated team and put them out there, you really can have a big impact on slowing this disease. But in order to get to that point, you really have to scale things up. So ideally, we would have several of these teams working across the Caribbean at places that are impacted by SCTLD. And there are some now, particularly in US Virgin Islands, in Bahamas, etc. So we also wanted to try to understand, can we better optimize how we're intervening on these corals by looking at changes in gene expression. So just like us, corals turn on and off different genes in response to getting an infection, getting sick. And it was not a surprise at all when we did an ex situ transmission where in an aquarium setting, we artificially infected some corals down in Miami with a former postdoc and grad student of mine, Mike Sedevin. And we found, not surprisingly, that getting this disease dramatically alters coral physiology. But what was interesting and connects to those same results from the field work about the Orbicella is that individuals and different species respond differently to being infected with these corals. They turn on different sets of genes. And what was most interesting is that when we then compared how these diseased corals looked after we treated them with the amoxicillin, what we found is that once those corals were treated with amoxicillin, their gene expression profiles essentially returned to those of healthy, normal, non-diseased individuals. So the amoxicillin is not only stopping the lesion, it's also kind of returning the corals to a more, quote, normal functioning physiology than they were before. So one approach would be to do intervention, but then what do we do about all the corals that we've already lost? And there's been an increasing push here in Florida and throughout the world to do more active coral restoration. That is developing coral nurseries, growing up corals, and then outplanting them back onto a reef in the hopes that they'll recover. However, given the risk of stony coral tissue loss disease, there was a lot of concern that all we'd be doing is putting corals out on the reef and sending them to a death sentence because they're going into an active disease situation. So to assess the relative risk of getting infected during a coral restoration experiment, we partnered with FWC, University of Miami, NOVA, and Mote Marine Lab to conduct what we think is the largest coral restoration experiment in Florida to date. So we outplanted a little under 6,000 coral fragments in batches of 5. And so all 5 of the individuals, like you see here in the center, are pieces of the same coral genotype, the same coral individual that have been allowed to grow back and fuse together. So we set up arrays of these coral restoration plots in 6 different regions along the coast of Florida, with 4 different restoration plots, each containing 48 little colony units like this throughout the region. So a whole lot of corals have been put out on the reef to see how they will respond with 3 different coral species. So we've been monitoring them very intensely, originally weekly and then monthly after month 1. And long story short, these corals have done really well. Very strong survivorship, as you can see there in that chart on the right, which is average % live tissue per colony over time. You can see that some of the sites might do a little bit better than others, but all of them are over 70% live tissue cover still. And the tissue cover that we have lost, most of that has been either due to an individual coral fragment being popped off, either by a fish biting it or getting lost during a high wave event, there's really been very minor disease impacts, particularly in our region, up in Region 1, which is Martin and Palm Beach County. So at least so far, it appears that coral restoration with these species, of rain coral and 2-star corals, can effectively move forward without an increased risk that all of those individuals are just going to die from this disease. Our next steps are going to be to try to understand the relative performance of these corals across all 6 regions. Gabby will be looking at a whole bunch of image analyses to understand coral survivorship and growth over time. And Sydney, another grad student in my lab, will be looking at the genotypes and the algal symbionts within these corals to see if we can predict their relative success, or if there's correlations between success in a particular region and the genotype or the symbionts associated with that coral. Now, here in South Florida, we also know that water quality is a huge issue. And so we hypothesized that perhaps water quality might be driving some of the increases that we were seeing in disease in Southeast Florida. During my graduate work, I had found that excess nutrients could cause an increase in diseases in a different disease called black band disease. So we wanted to test that on stony coral tissue loss disease. And so this is part of Ashley Carrero's thesis. Basically, she took a bunch of naturally occurring disease infections, and half of them got dosed with nutrients and an ecologically relevant amount of time-release fertilizer. Half of them were controls, and then she also had an additional 15 that were apparently healthy individuals that had no signs of disease. And she tracked the relative rates of progression of the disease over time using both 3D modeling with photogrammetry and linear progression. And then she also recorded nutrient concentrations to verify that giving them fertilizer was actually increasing the amount of local nutrients. So she'll be looking at whether or not these nutrients are actually causing an increase in disease. We're not sure that it does, to be perfectly honest, so far. And also whether or not there's any changes to the coral microbiomes that result from poor water quality and excess nutrients. And the idea behind this one is to try to provide recommendations that can help to set threshold criteria for what amount of nitrogen or phosphorus corals can actually handle when they're being impacted by disease on a reef. The other part of water quality that we're interested in is salinity. So I've been working with a broad team trying to understand what kind of criteria and performance metrics we need to better manage water flow in Southeast Florida. So the Lake Okeechobee monitoring system is going to again be revised, and the operational criteria are going to be reevaluated and are being reevaluated right now. And what we were quite surprised to see when we went to try to understand, well, what salinity thresholds can corals handle, there was really not great data on that for corals in Southeast Florida. So student by Haley Davis is currently conducting a project to try to identify 3 different things. Number 1, whether or not we can identify kind of the acute stress threshold points for salinity. At what point do these corals quickly tank once the water starts to get too fresh? Number 2, we want to look at a moderate level of low salinity stress, if that's extended for a long period of time, how long before the corals start to essentially die from that? And then lastly, if corals are exposed to salinity stress, does that increase their susceptibility to disease? So we're going to stress out some corals with salinity stress and have a control group that's not salinity stress, and then expose both of them to disease to see if there's a differential susceptibility to infection. So we've got a lot going on, and we've accomplished a lot so far. Things I'd like you to remember for today is that number 1, coral reefs really are kind of ecological and economic powerhouses. They're incredibly important to our overall ocean function and to our local economy here, but corals are under severe threat and need increased effort and management in order to continue to provide the things that we benefit from. As we've looked more, we're finding these mesophotic reef habitats truly are extensive and appear to be relatively well connected across broad spatial scales, from Florida to the Gulf of Mexico to Cuba, even down into Belize, and that we hope these should provide a source of recovery for shallow reefs. It also means that it's important to protect these mesophotic habitats so that they're not degraded and they can continue to provide a potential resilience and recovery for other areas. We know from experimental work as well as ongoing efforts in the field by several groups that we can use antibiotics to do direct interventions on corals and save those corals that have active disease. But it's really unclear at this point whether or not we can scale up those efforts to have ecological effectiveness. Can we do enough intervention that a reef can be saved, not just a coral can be saved? And so there's some ongoing work in both NOVA and Dry Tortugas National Park to try to address this issue as well. I think that coral restoration research is necessarily going to have to expand. We've lost too many reefs, and if we want those ecosystem functions back, we're going to have to do coral restoration. But I think there's still a lot of questions about what is the best way to do coral restoration. And I think that there's several different groups that are experimenting on this front, and that we need to have good rigor across big spatial scales with lots of species like we've done in that recent work with FWC to really give ourselves the blueprint of what could be effective going forward. And as a result of a lot of the exploration that we've done and data that we provided, new MPAs have been created, sanctuaries have been expanded, and there's revised management plans that have been achieved in multiple regions. So it's my quick way of saying it's worth it. That if we do this kind of work, we can move the needle in terms of management and hopefully protect areas before they start to degrade beyond recovery. So one of the other things I wanted to add was some recommendations that things that you as an interested audience can take on or discuss or even do yourselves. I think as a group of scientists, we're going to have to support and pursue really innovative and effective conservation and restoration practices. We need to fund and support some of these high risk, high reward kind of approaches. Several different areas within the government are starting to be supportive of that, as well as several foundations as well. So things like genetic manipulation of corals, assisted evolution, green-gray infrastructure where you combine artificial reefs with living corals to try to do restoration and to sustain coral ecosystem services. All these things are underway and should be supported. I think we really need to expand our regional and global scale collaborations. The more we do this connectivity work, the more it demonstrates to me that corals do not respect our national or even MPA boundaries, obviously. And so we need to build these partnerships across broad space in order to effectively manage the entire resource, not just a piece of the resource, in a more integrated way. Here in South Florida, supporting enhanced water quality management and infrastructure. In other words, biting the bullet and doing more conversion from septic to sewer, improving sewage treatment plants so that discharge water is cleaner before it goes out, are all things that need to happen, as well as efforts to address climate change, since we know thermal stress adds an additional stress to corals as well. I think that we're starting to be able to advocate for developing a management plan. So here in Southeast Florida, we have the Coral Reef Ecosystem Conservation Area, which essentially is Florida's newest MPA. It's the Kristen Jacobs Coral Reef Ecosystem Conservation Area that essentially runs from the northern edge of Biscayne National Park all the way up to Martin County. And now that that box has been drawn, we need to develop a management plan for it. So as residents of this region, advocating for that management plan and supporting that management plan is something that's going to be important for all of us. we need to continue to not only train, but inspire people to want to go into coral reef research and conservation and to be ocean stewards. So I think investing time and resources in giving people the opportunity to get underwater, to get at sea, can really help to inspire people to want to pursue this field and help with coral conservation. And then lastly, really the telepresence work that we've done has demonstrated to me that we have to embrace storytelling as our best way to enhance broad education and outreach. Data is still needed in order to implement management plans that can be adaptive and effective. But when you're trying to build that groundswell of support for a management plan, the storytelling is often much more important than the data. So teaching those of the next generation to be great storytellers is important as well. And with that, I'll say thanks to everyone. I'll be happy to answer any questions that may have popped up. I'll leave you with a hope image here. This is corals spawning in Flower Garden Banks Sanctuary that we were able to record and broadcast live. So you can see that some of these corals are releasing clouds of sperm, others are releasing packets of eggs that are going to fertilize and spread throughout the Northwest Gulf. So some areas indeed are doing really well. These are the areas that we should try to protect the best. Thanks all.
Thank you, Joshua. And just as a reminder, if you have any questions for Dr. Voss, please write that down under the Q&A button in the bottom of your screen. Joshua, you mentioned that, and it sounds very much like the amoxicillin treatment is very tedious if you have to go down there and treat basically individual corals. And you mentioned that more widespread treatments are needed. Do you have any idea on what that would look like to do more widespread treatments rather than the individual amoxicillin treatments?
Well, I definitely, when I say more widespread and scaled up, I don't mean spraying antibiotics across the reef. I think that's rather irresponsible. Yeah. But we know that we need to have these targeted releases. So that base 2B that we use is designed to be quickly time released. So essentially it holds onto the antibiotic and releases it onto the coral, but then it's basically gone in just about 3 days. The reason that we chose amoxicillin is that amoxicillin is something that's already in local watersheds and in local coastal communities from sewage runoff. So we didn't want to introduce a new antibiotic into our coastal ocean systems. We wanted to use one that was kind of already present there. However, I totally agree that we need to worry about antibiotic resistance as well. And so we have, as part of the projects that we've done, we've sampled the microbes to screen for antibiotic resistance genes. And there's several other projects that are looking at this potential as well. The results we have so far suggest that yes, it's effective. There are not subsequent negative effects to other organisms in the nearby vicinity. So within an area around 3 meters of these treated corals, there's no changes in the healthy corals, there's no changes in the healthy sponges, there's no changes in any of the other benthic organisms that are associated in that area. So we think that this approach is giving us a targeted dose of antibiotics. And again, when I say scale up, I mean more people doing these applications, not spraying antibiotics out the back of a boat or an airplane.
You kind of answered the next question with this, which is related to the possible negative effects of having widespread use of antibiotics, right? And what the potential negative effects could be if you're treating a human with too much antibiotics, they will die too. It's always a dose effect, right? So is there any knowledge on how much is too much?
So the doses that we've been using that are effective, none of them have caused death on, so we've also, sorry, I say we, the royal we, Cindy and Karen have trialed this antibiotic on both diseased individuals and healthy control individuals to see whether or not there were any negative impacts of applying the antibiotic to a healthy individual. And they did not see any effects, any negative effects the healthy individuals of getting treated with antibiotics. So that suggests that at least at the concentrations we're using, the antibiotics aren't innately bad. But in terms of, can we put too much on the reef that it starts to change things? I think potentially. I don't think any group is putting out enough that we need to be concerned yet. But if we were to all of a sudden have thousands and thousands of volunteers that were willing to do this, I think we would first need to do a study to make sure that we're not artificially increasing antibiotic concentration in the water. Those that have been done so far by oceanographists do not demonstrate that there's an increased antibiotic concentration further than about 30 centimeters away from the treated corals.
Now, the corals are used as homes for some fish. Is there any knowledge on whether the antibiotics get to the fish and affect the fish health?
I don't know yet. Though it would potentially be a problem for small fishes like, cleaner gobies and neon gobies that are truly resident on an individual coral. And again, the question of what's worse, that they're briefly exposed to antibiotic or that they lose their home is an open thing to discuss and to try to tease out.
Yeah. Okay. One more question here from one of our participants. Has anyone looked at septic tank leakage, whether that might be causing these diseases? The suggestion here is because antibiotics are helping, that might be indicative of a potential link.
So the fact that antibiotics are working suggests that this might be a bacterial infection, right? But there's also some microscopic evidence that there could be a potential virus-like particle involved as well. And again, that virus particle could be a virus that's carried by a bacteria, so it could be incredibly complicated. However, there's not been direct studies of taking septic effluent and trying to see whether or not that causes SCTLD lesions. There has been some correlative studies where Gareth Williams and Brian Walker have looked at the relative distribution of diseased lesions and diseased corals across Southeast Florida, and then tried to look at, okay, what's related to this? What are they close to? And there is a very weak but significant correlation between septic tank density, or the number of septic tanks within a region, and the prevalence of coral disease within that region. But again, it's significant, but it only explains a small fraction of the variance in the data. So I think that there's strong evidence that septic tanks cause all kinds of disruptions in estuaries. There was very strong evidence of septic tanks causing problems in the Keys long before starting coral tissue loss disease. So I think there's already a lot of evidence of why septic tanks are not good for corals, but there's not yet a direct test that has shown whether or not septic tanks are directly leading to SCTLD.
Thank you. We're about out of time. So I want to thank you, Josh, for your very interesting presentation. If anybody has additional questions, please reach out to Dr. Voss. And also, if you want to hear more on how you can help, how we all can help in the community, please feel free to reach out to Dr. Voss and our Harbor Branch community. With that, thanks again. I hope you all have a great rest of your day. Bye.
Thanks, Karen. Thanks, everyone.